Hello, everyone. This is Dr. Garrett Smith, the nutrition detective of nutritiondetective.work and nutritionrestored.com. And today I have a presentation that we're going to go over where I'm going to show you the research that demonstrates that what we call vitamin A deficiency doesn't exist. Let me get into that presentation right now, and then we will get going. Okay. Here we go. So, moving on, what we will be covering in this video, the types of research studies that we prioritize and prefer, the supposed signs and symptoms of the so-called vitamin A deficiency state, animal studies debunking the so-called vitamin A deficiency, which we will be abbreviating vitamin A VA for short. We have human studies debunking vitamin A deficiency, and we have nutrient deficiencies that cause the exact same problems as vitamin A deficiency, and that was the mistake that they made. And some current research going on in humans and animals on low vitamin A diets. So, moving on. These are my priorities in looking at research on the so-called vitamin A. The ones that we consider much less valuable are animal studies, because they're not humans, short-term studies, because long-term is really what we're after in terms of long-term health. In vitro studies, which some people call petri dish studies, mechanism studies, where they're just looking at theoretical ways they think things are working rather than looking at the real world output, and blood test only studies where they're looking at blood test values and not actually the actual function. The ones we consider much more valuable are kind of the opposite of those. We want human studies, we want long-term studies, we want in vivo, which means in a living organism, not just in a petri dish kind of thing. We want signs and symptoms studies where they look at what's actually happening in the organism, in the person, and actual human function observation studies, rather than saying, you know, they have low vitamin A on a blood test, but they don't talk about problems. We want to know, do they have actual problems? So these are the supposed classic signs and symptoms of vitamin A deficiency. These are what they were believed to be. We have ocular symptoms, or the eyes. We have poor dark adaptation, what a lot of people call night blindness or poor night vision, also known as nyctalopia. There is the dry eyes disease or syndrome, which is also called xerophthalmia. This disease, once it goes along a certain distance, turns into keratomalacia and is somewhat irreversible. And then we have macroscopic, larger, and microscopic, stuff you can only see under a microscope, changes to the eye tissues. We have dermatological or skin issues said to happen. Macroscopic and microscopic changes in the skin, stuff you can see with your eye and stuff you have to see with a microscope, also known as various skin diseases. And then the hematological changes, blood changes, these can include changes in white blood cell counts. Various ones, some of them go up, some of them go down. These are what supposedly vitamin A deficiency does. So, yes, moving on, we have animal and human studies. The first paper, and these links all link to either the study itself or a reference where you can find it. The slideshow is linked, is accessible and linked in the notes below this video. So if you want to see the references, these are all quotes. When I quote, I'm quoting the paper. So we have the first one, Pathogenesis Due to Vitamin Deficiency in the Rat by Emmett and Allen. They used to call it vitamine with the extra E. They dropped that along the way. So first, quote, the rats were fed on definite dietary planes which would produce normal growth except for the lack of either vitamin A or B. From these groups, animals were selected which were representative. Care was taken to see that the animals were free from infection. Control tests were made on tissues from rats fed a complete diet. In the rats which lacked vitamin A in their diet, there were no special outstanding pathological findings. In marked contrast with the lack of vitamin B, the liver showed no fatty changes, the adrenals no hypertrophy, and the thymus no atrophy. The control animals proved to be normal as far as histological examination showed. So what they're saying here is that in the animals that didn't get vitamin B, and when they mean B, they hadn't at this time broken the Bs out into all the separate B vitamins like we know today. they were kind of talking about vitamin B complex as a whole, as we know it now. But what they're saying is that in the vitamin B deficient rats, they saw fatty livers, they saw hypertrophy of the adrenals, and they saw atrophy of the thymus. And they saw none of these changes in the control rats or the rats that got no vitamin A. Okay, so there's our first paper. Moving on, I want to go into, do humans need carotenoids at all? And if you're not familiar with what carotenoids are, beta carotene as an example is called a provitamin A carotenoid, which means that your body will cut one beta carotene into two retinols or retinaldehydes. It doesn't turn it into retinol. It turns it into retinaldehyde. And then your body, through the various metabolic processes, can move it backwards into retinol or forwards into retinoic acid. I go into all this stuff about vitamin A metabolism in the detox program. But from this paper, 2013 review paper called Carotenoids, quote, none of the carotenoids are considered essential nutrients. No carotenoid is directly involved in a vital metabolic pathway, nor has the relative absence of a carotenoid been linked exclusively to the induction of a specific deficiency or chronic disease. Hence, the lack of FDA-approved health claims, end quote. All of the retinoids in your body, regardless of what some of the people out there on the internet are saying, that your body doesn't turn carotene into retinol and all this stuff, all retinoids, whether they are in us or whether they were in the animals that we eat, they all are breakdown or degradation products of carotenoids. There would be no retinol or any other retinoids in the body of you or the animals you might eat without carotenoids to start with. None. All vitamin A comes from carotenoids. So if this base product of the so-called animal vitamin A, the one that everybody says is essential, is entirely rooted in carotenoids, and humans can convert carotenoids, we can break them down into retinaldehyde just fine. There's lots of studies that show that. And if carotenoids are not essential in any way, wouldn't this alone seriously call into question the essentiality of all vitamin A compounds by itself? Moving on. Paper called Vitamin A Deficiency and the Requirements of Human Adults. Quote, 23 conscientious objectors, 20 men and 3 women, volunteered to live on a diet designed to be deficient in these substances but complete in every other respect. The absence of vitamin A and its precursors was confirmed spectrophotometrically and biologically. The disappearance of carotene from the blood plasma of the deprived volunteers confirmed the absence of these substances from the diet. With efficiency for dark adaptation and plasma values for vitamin A as criteria, none of the 16 deprived subjects became depleted within a year. They knew that the diet had no vitamin A in it. They watched their blood values of carotene go down. They did not see any changes in night vision over a year's time. Do you not think that if this was a real vitamin and if we really needed it, that a year without it would probably show symptoms? What I talk about a lot is vitamin A toxicity, which means that we are accumulating this poison in us. And that is what happens. None of them got depleted within a year. Moving on. Vitamin A and epilepsy, a dietary contretemps. I looked up how to pronounce that. I hope I did okay for you French speakers. I took this summary from another paper because I couldn't find the original paper anywhere online. But lots of papers quote this paper. So here we go. Quote, in order to investigate further a possible connection between vitamin A and epilepsy, Sharman then studied 8 epileptic patients given a vitamin A depleted diet for 2 years. Not just 1 year now, 2 years. No cases of night blindness were observed. When their plasma retinol level had reached 230 international units per liter, which is not the measurements we use today, so don't compare it, they were given a vitamin A supplement. Although the patients reported fewer fits during the depletion period, there was no increase in fits during the repletion. The author called his experiment a dietary contretemps. First of all, what's the definition of a contretemps? It is an inopportune or embarrassing occurrence or situation, an unforeseen event that disrupts the normal course of things. The author went into this expecting that vitamin A would be necessary. The author, let me tell you, Sharman did this study because he had somebody come, or he heard of this case, or he had somebody come to him as a physician who this gentleman had decided that he was not going to eat vitamin A because his interpretation of the literature was that vitamin A was what caused epilepsy. This gentleman did his own diet. We don't know what was in this one person's diet. He had one epileptic fit after he decided to adopt a vitamin A-free diet. Okay. Then we have Sharman continue. He decided to do a study on this. 8 epileptic patients, no vitamin A for 2 years, or very little vitamin A. There is actually no such thing as a zero vitamin A diet. There's no such thing. Don't let anybody ever tell you that. 2 years, there was no night blindness, fewer epileptic fits. They decided that they were scared of these people getting vitamin A deficient. So when their vitamin A levels got to a certain point in their blood, they gave them vitamin A again because they were so worried of something happening that wasn't happening, right? So anyway, he was embarrassed that he was basically showing that vitamin A was not necessary and that less vitamin A was good for epilepsy. Interesting. Next, we have huge vitamin A. Sorry, I skipped one. Vitamin A requirement of human adults. An experimental study of vitamin A deprivation in man. Quote, the great majority of clinical examinations revealed no significant differences between the deprived and a non-deprived group or in the exact same person before and after deprivation of vitamin A. In several of the subjects, follicular hyperkeratosis was present at the start and varied during the experiment, but the variations bore no relation to the vitamin A intake. Same person before and after vitamin A, no difference, nothing worse. 2 groups of people with some with vitamin A, some without, no difference. People with skin issues, it went up and down like normal, but it didn't bear any variation with the vitamin A intake, which if you believe the people who think you need vitamin A for your skin, obviously this would kind of throw a wrench in that. So moving on. There has been research showing that there have been huge vitamin A deficiency populations by blood test without any eye problems. Did you know that? Here's the paper. Vitamin A deficiency in Latin America and the Caribbean, an overview. This was back in '98. Quote, vitamin A deficiency has been known to exist in Latin America and the Caribbean since the mid-1960s. There was little interest in controlling it because of the low frequency of clinical findings. In most countries of the region, however, vitamin A deficiency was not considered a problem of public health significance because ocular eye lesions leading to permanent blindness were not frequently observed, despite prevalences of low serum retinol, aka vitamin A, hovering near the cutoff of 15%, then recommended by the WHO, the World Health Organization. On the other hand, ocular signs attributable to clinical vitamin A deficiency were extremely rare in the 1980s, and they have not been assessed more recently. Could you get any more obvious than this? Giant populations who are testing low in this supposed vitamin. Nobody has problems that they would expect. Where are all these massive problems? Why aren't they all having massive skin issues and eye issues? Maybe because it's not a vitamin. Okay. Next, another study where we have more vitamin A deficient people by blood test with no symptoms. The paper, Retinol Deficiency and Urinary Stone Disease. Clinical evidence is missing. Interesting title. Quote, it was found that 68% of subjects in lower socioeconomic group had serum retinol, vitamin A, levels between 10 and 19 microgram percent, and 4% below 10 microgram percent, but none of them showed any symptoms of retinol deficiency. So that's 68 plus 4, that's 72% of these people in this paper. None of them had any signs of retinol deficiency, even though some of them were severely deficient based on their blood tests. This is really important. Now this is a fun one on kids, because people say, oh, you need vitamin A for development, proper development, you need it. From the Food and Nutrition Board of the Institute of Medicine, the IOM, this is a big US government organization. From their giant book, Dietary Reference Intakes for Vitamin A, Vitamin K, Arsenic, Boron, Chromium, Copper, Iodine, Iron, Manganese, Molybdenum, Nickel, Silicon, Vanadium, and Zinc. Quote, the mean liver vitamin A stores in apparently healthy infants is lower, ranging from 0 to 320 micrograms per gram of liver. This is when they say 320 micrograms or 0 to 320 micrograms per gram of liver, this means they had children who had passed, whether by disease or accident, children who had died, and their bodies were donated to science. And they took a liver biopsy, they took a chunk of their liver, and they analyzed how much vitamin A on it. So they're saying they were apparently healthy at the kids who died here. These were probably accidents, is what I'm guessing for this paper, because they were apparently healthy. They were not diseased, yet they died. So like think car accidents, stuff like that. So they had apparently healthy infants with 0 vitamin A in their liver. 0. This is the gold standard test of vitamin A, folks, the liver biopsy. The gold standard. If you have 0 vitamin A in your liver, that pretty much means you have 0 vitamin A in your blood. How is it possible that these infants were healthy if vitamin A is supposedly critically necessary for proper growth and development, and they had none? There is only one answer to that, and that is that vitamin A is not an essential nutrient. It's not a vitamin. Here's the first big study. The paper is called Effects of Vitamin A Depletion in Young Adults. So what are they going after here? Lowering vitamin A in people? Quote, the lack of agreement as to what constitutes subclinical vitamin A deficiency and the uncertainty existing as to the order of the occurrence of the supposed early signs in relation to one another prompted the undertaking of this study. The various suggested symptoms of early avitaminosis A include defective dark adaptation, macroscopic and microscopic changes in the skin and conjunctivas, that's in your eye, changes in the total and differential white blood counts, and decreased vitamin A content of the blood. We would expect that, right? Each of these factors has been investigated individually. There have been studies in which only 2 or 3 of the aforementioned signs have been followed in any one subject, the most usual method being to observe dark adaptation and one other sign. It was believed, therefore, that careful observations for all of the suggested subclinical signs of progressive vitamin A deficiency in the same subjects on a vitamin A low diet would at least clarify the order of their appearance. End quote. Do you understand how much confusion they're saying is going on there? Some of the key words: lack of agreement, uncertainty. What else do we have? We have clarify. There's all sorts of stuff going on in there. They're saying suggested symptoms. That was the one I missed. They're saying this doesn't make any sense. This is not working the same in all these papers. So let's continue going on. The experimental methods. Quote, 6 university students, 4 women and 2 men ranging in age from 20 to 24 years were placed on a vitamin A low diet for 31 and 20 weeks respectively. 31 weeks, this is my addition, 31 weeks is 7.5 months. 20 weeks is 4.5 months. Continuing. The diet contained from 67 to 100 USP units of vitamin A per day. That's very low. Continuing the quote. The method employed for detecting the effects of vitamin A deficiency included measurements of dark adaptation in visual fields, of the vitamin A and carotene content of the blood, and of histologic changes in the skin and conjunctivas. Total white blood cell counts and differential counts were also included. Most of these analyses were made prior to the experimental regimen so that the normal baseline for each subject could be determined. So they had numbers on these people, end quote, but there was the end quote. Most of these people had numbers on these things before they went into the study. That's really important. They had a baseline before they went on the low vitamin A diet. So we should really know what changed, right? Number one, dark adaptation, section 1. There's a couple of these for each section. So, quote, biophotometer, a comparison of the 20 to 30 second dial readings followed by bleaching of the 6 subjects with similar readings reported by genes, places 5 of the subjects in the borderline zone and 1 in the subnormal zone. The deficiency diet had little effect on these readings. Although there were slight fluctuations in both directions, i.e. from borderline to normal and subnormal, the subjects maintained their relative groupings, end quote. So they didn't get worse. They stayed the same. Part B of dark adaptation, the adaptometer, quote, the cone and rod thresholds for all subjects fell within the normal range reported by Hecht. Throughout the entire period, those thresholds remained constant for each subject, fluctuating only within the daily variation of 0.3 to 0.4 of a log unit for rod and cone thresholds, respectively. In figure 2 are presented the cone and rod thresholds for every test on subject FW. The data for other subjects show similar figures. They did have one subject that they gave vitamin A to the whole time. They gave him like 10,000 units so they could see what happened when they supplemented. I don't remember, but FW might be the one who got the vitamin A. And they're saying everybody else showed similar figures to the person who got vitamin A. Part C of dark adaptation, regenometer. The time necessary to see the 5 consecutive flashes of the first and second dot for time, I think it was. I think when I copy and pasted that, it must have changed the I and the M into the subject was well within the normal time limit given by Blanchard, namely 1 and 3 minutes respectively. Part D, visual field. The visual field did not seem to be impaired in any way as measured by perimetry. So in all of their dark, I'm positive that was time up in that quote earlier. I just missed that cut and paste change. All of the dark adaptation, no changes, nothing. We had multiple papers earlier where they said there was no change in dark adaptation. The Latin America and Caribbean said there was no night blindness going on. Part 3 of this paper, the blood picture. Part 2 was going over the fluctuations in vitamin A. For those of you who really want to understand how this all works, I went over this recently in my inner circle group on the vitamin A detox group, how these things go up and down. And I can explain all of the ups and downs that they had. When you understand this, it all starts to make sense. When you understand it's a poison and coming out. The blood picture part. Quote, according to the reports of Abbott and Associates, the blood picture should be a valuable aid in the diagnosis of vitamin A deficiency because of the marked changes they obtained in rats and human beings on a vitamin A low diet. Namely, a decreased number of total white blood cells in polymorphonuclears or heterophils, and an increased number of lymphocytes with the large lymphocytes predominating over the small. In addition, these authors, they're talking about the past ones, found many juvenile and stab forms and degenerate cells. None of the subjects in this study showed this type of picture, end quote. No changes in their white blood cells based on what the previous authors were stating was what vitamin A deficiency would do to the blood. Part 4, skin. Quote, the biopsies made at 4 and 7 month intervals of deficiency revealed no measurable differences in the subcutaneous structure of the hair follicles and sweat and sebaceous glands from that found prior to the experimental diet. The visibility of the capillaries remained as good as at the start, demonstrating that there was no keratinization of the epithelial tissue at the region studied, end quote. Wow, this is not going good for what they were looking for, is it? Examination of the eyes. Wow, this should be a big one. Quote, at the last week of deficiency, there were no gross ocular signs, end quote. Part 6, subjective signs. Quote, the general health of the subjects throughout the experiment was as usual. There were 1 or 2 colds during the winter, but this was no more than in previous years. There were no reports by the subjects of subjective night blindness, end quote. The comment section of the paper. Quote, it is seen, therefore, that normal, well-fed adults did not present any of the alleged signs of subclinical vitamin A deficiency even after 7.5 months on a depletion diet, end quote. From the summary section, 5 young adults, 3 women and 2 men, were placed on a vitamin A low diet for 7.5 and 4.5 months respectively. At suitable intervals, measurements were made of dark adaptation, blood vitamin A and carotene, total white blood cell counts, and differential blood counts and changes in the structure of the skin and conjunctivas. None of these measurements showed any definite changes from those found prior to the deficiency for the length of time studied. So we now have papers a year, 2 years, 7.5 months. Now we go into our second big study. This is a rat study, but this is really, really important. The pathological anatomy of ophthalmia, that's eye disease, produced by diets containing fat-soluble A, but unfavorable contents of certain inorganic elements. So this first slide is to make sure we all understand what we're talking about here. Inorganic elements are minerals. Salts are combinations of inorganic elements, like sodium chloride. So the essential inorganic elements or minerals, here's a list. There's probably a couple missing here, but this is the list that I used. Zinc, selenium, molybdenum, magnesium, potassium, chromium, phosphorus, calcium, copper, chloride, magnesium, phosphorus, potassium, sodium, chromium, cobalt, iodine, iron, manganese. These are the minerals that the human body needs to function. Okay? What we're about to cover in this study is specifically stating, they will show and state that the exact same eye problems said to be caused by vitamin A deficiency are also caused in rats by diets containing plenty of vitamin A. But those vitamin A containing diets had unfavorable contents of minerals. Now, unfavorable contents could mean too much or too little. Okay, so now we're going to find out about why these studies don't agree. The ones that say vitamin A deficiency is real and the ones that say it's not, why they don't agree. Quote, recently, McCollum, Simmons, and Becker have reported that diets containing an abundant supply of fat-soluble A may produce an ophthalmia in rats. Their diets contained different salt-mineral mixtures. Moreover, they seem to resemble each other only in that they had a high content of sodium and chlorine, AKA that's table salt, sodium chloride, okay? The disease is very similar clinically to that which develops in animals which are fed diets deficient in fat-soluble A, end quote. So that when they're talking about McCollum, Simmons, and Becker, they had rats on a diet that had plenty of vitamin A, and then they had rats on a diet that had none. But the two different groups of rats got different minerals, okay? The only thing that these diets had in common was that they had a high content of basically table salt, sodium chloride. But here's the weird thing, because people will try to say that the eye diseases of vitamin A deficiency are what they call pathognomonic, which means that they are indicative almost automatically and perfectly of vitamin A deficiency, okay? These diets produced, quote, clinically and pathologically identical eye diseases, okay? It's really important. Continuing from quoting the paper, xerophthalmia, dry eye, which is dry eye, and keratomalacia, which is the untreated progression of xerophthalmia, are produced in rats by feeding them diets which are deficient in fat-soluble A. The same changes may result from consumption of diets which have, I believe that's pronounced ceteris paribus, which means all things being equal, an abundant supply of fat-soluble A, but which contain certain unfavorable salt-mineral mixtures. Here's the really important part. The two conditions are clinically and pathologically identical and are identical with human xerophthalmia and keratomalacia. It would seem from the foregoing experiment that in cases of xerophthalmia which occur in human beings, other factors other than fat-soluble A may have to be considered in regard to the etiology and treatment of the condition. End quote. Let me read that last sentence again, because these authors figured it out. And this paper, I want to say, they're still talking about fat-soluble A. This has got to be from the 1920s, okay? It would seem from the foregoing experiment that in cases of xerophthalmia, dry eye, which occur in human beings, other factors other than fat-soluble A may have to be considered in regard to the etiology, the cause, and treatment, how you fix it, of the condition, end quote. Here's a quote. One rat in which keratomalacia had been produced was placed on a diet containing a good salt mixture, good mineral mixture, after both cornea had been perforated by ulcers. The activity of the pathological process was checked at once clinically as a result of the change. Photophobia disappeared, the discharge diminished, and the conjunctiva became moistened, end quote. What are they saying there? When they didn't say this rat, they didn't say whether this rat was on a vitamin A diet or whether it was on the wrong salt mixture, the wrong mineral mixture diet. They didn't say. So they gave this rat ulcers in its cornea, okay, perforations, holes in its cornea with whatever diet they gave it. After this happened, they gave the rat the good mineral mixture. The process stopped immediately and started reversing. Photophobia means they don't like bright lights. If you didn't know, vitamin A toxicity, a big sign of it, an easy sign of it in people is you don't like bright lights. So they're saying the photophobia disappeared, the discharge, the gunk coming out of the eye stopped, and the conjunctiva, the dry eye, became moistened. It became wet. It all went away when they gave it the right mineral mixture. Huh. What would that last thing say? It might be caused by other things. So now we're going to get into the other essential nutrient deficiencies that are causing what are supposedly vitamin A deficiency problems. One of these nutrients that causes what people think are vitamin A deficiency signs and symptoms is zinc. Let me read this from the paper, Zinc and its Deficiency Diseases. Quote, symptoms resulting from zinc deficiency are as diverse as the enzymes with which the metal is associated. If chronic, severe, and untreated, zinc deficiency can be fatal. This is important for later, so pay attention to that part. Less drastic symptoms include infections, hypogonadism, weight loss, emotional disturbance, dermatitis. That means skin irritation, skin issues. Alopecia, impaired taste acuity, night blindness, poor appetite, delayed wound healing, and elevated blood ammonia levels. Many symptoms of zinc deficiency result from poor diet consumption, but often the most severe symptoms result from other factors, including excessive alcohol use, liver diseases, malabsorption syndromes, renal disease, enteral or parenteral alimentation, administration of sulfhydryl-containing drugs, and sickle cell diseases, end quote. So we've got eye problems. There's a lot more eye problems to zinc deficiency than just night blindness. We got skin problems, dermatitis. Dermatitis is what starts and then it turns into other things. And there are other skin issues related to zinc deficiency. I couldn't make this presentation go on too long. Now, they said fatal. Well, this is going to be really, really, really important when we get to the Wolbach and Howe study later. So remember, zinc deficiency can kill. Okay, moving on. Now we're going to go into taurine, which is said to be by many researchers a conditionally essential nutrient, which means that under certain conditions, you may need it from your diet more so than your body. Your body can make some taurine, but under certain situations, you need more than your body can make, which makes it an essential nutrient, okay? So this is from a book called The Biology of Taurine, Methods and Mechanisms. So first, I want to get into what is a photoreceptor, because they're going to talk about it. A photoreceptor is a nerve-ending cell or group of cells specialized to sense or receive light. Light, that's what it means. So, quote, there is now little doubt that taurine is involved in the mechanisms preserving the structural and functional integrity of photoreceptor cells in the vertebrate retina. The supportive evidence comes from one, in vivo experiments in taurine-deficient animals in which the structural and functional disturbances occur in the retina following a decrease in taurine retinol levels. Really what they meant to say there, I'm going to stop there. They meant to say retinol taurine levels, because one of the confusing things they did in medicine was they said retinal, which is retinaldehyde. I don't know if you know this, but one of the forms of vitamin A in your body is retinaldehyde. It's an aldehyde. Aldehydes aren't good, if you didn't know. If you didn't know that one of the forms of vitamin A as it moves through your body is an aldehyde. Retinol is an alcohol. Retinaldehyde is an aldehyde. And then you have retinoic acid, which is an acid. And then you have all the people out there saying acid alkaline diet. Are you seeing alcohols, aldehydes, acids? None of those are vitamins. Anyway, there will be retinol taurine levels, because they mean referring to the retina and the taurine levels in the retina. And they have 4 references there. So decrease in taurine retinol levels in vitro experiments in preparations of isolated photoreceptor outer segments where taurine affords protection against different experimental conditions causing a disruption of the highly organized structure of the outer segments, end quote. What they're saying is that taurine is necessary for night vision. Okay. Is there more to taurine? Well, let's go into the paper called Antioxidant and Osmoprotecting Activity of Taurine in Dry Eye Models. Quote, the purpose, to assess the protective effects of ophthalmic formulations based on taurine and sodium hyaluronate in ocular surface. Methods, rabbit corneal epithelial cells were subjected to oxidative stress and treated with the following formulations: 0.2% SH, which is the sodium hyaluronate, 0.4% SH, 0.4% SH plus 0.5% taurine. Dry eye was induced by atropine sulfate, and topical treatment was carried out with the following formulations: 0.2% SH, 0.4% SH, 0.4% SH plus 0.5% taurine. So I'm going to stop the quote here for a second. Only one of those formulations had taurine in it. Only one. Results. Taurine significantly decreased reactive oxygen species production in SIRC. I don't remember what that was. I didn't have a note here of what that was. After oxidative stress. All the altered parameters were significantly reduced by 0.5% taurine plus 0.4% sodium hyaluronate treatment. Furthermore, this formulation was more effective than sodium hyaluronate alone. So what did the authors conclude? Conclusions. Although altogether, these data suggest that taurine has a relevant antioxidant effect in corneal epithelial cells and prevents the ocular surface damage elicited by atropine, end quote. So taurine, not SH, not sodium hyaluronate, taurine prevented dry eye. Okay, weird. These both are symptoms of zinc and taurine. It's coming up again. Taurine has both night vision and dry eye. Here's a paper called Taurine, 2-aminoethane sulfonic acid, useful in skin diseases. Quote, taurine or 2-aminoethane sulfonic acid occurs naturally in food, especially in seafood and meat. Taurine is used in topical anti-acne therapy. It was used to explore anti-tumor promoting activity in a skin carcinogenesis. It is used in skin ulcers, Due to its antioxidant and anti-apoptotic properties, it exerts an anti-wrinkle effect on the skin. Taurine is effective in treating the underlying causes of psoriasis. It is also used in atopic eczema, end quote. Wow, that's a lot of skin problems. You ever heard about vitamin A being talked about with acne or wrinkles? I have. Does it work that great? No, that's why they have to give you more and more. That's why you see these commercials on TV where they're giving retinoic acids now and they're doing super retinol, and it doesn't work forever. It's like a drug. We'll talk about that. It does have effects. It's like a drug effect. But what I'm getting at here is taurine positively affects the skin. This is really important for later also. Both the zinc and the taurine connections here are really, really important. So that was taurine. We're moving on here to another paper, The Influence of Diet on Tear Function. Quote, the effect of diet on tear function is illustrated clearly by malnutrition-induced xerophthalmia, dry eye. Dietary habits in well-nourished North American society have been implicated as a cause of some tear dysfunction. A review of the ocular literature suggests that sufficient dietary protein, vitamins A, B6, and C, potassium, and zinc may be necessary for normal tear function. Excesses of dietary fat, salt, cholesterol, alcohol, protein, and sucrose sugar have been associated with or suggested as causes of tear dysfunction. So wait, a garbage diet and bad habits could cause dry eye. Lack of dietary protein could cause dry eye. And then we have those pesky inorganic elements, the minerals, zinc and potassium coming up. Okay. Hopefully you see from this paper that dry eye has been connected to all sorts of things, not just vitamin A. Okay. The supposed deficiency. So here's the question that people will say, or they'll try to, come back at this. They'll say, but research shows that vitamin A, and I'm just going to say here in general, does stuff. Vitamin A does stuff. Okay. First, you must understand the difference between essential to function normally and a compound having a drug effect or a biological effect, which are often similar. When you're talking about taking things, you're talking about either a drug effect or a biological effect, and the two are the same thing. Drug effects can be biological effects, but biological effects are not necessarily drug effects. Some examples: coffee, a natural substance, contains caffeine, which has the drug/biological effect of temporarily increasing energy. But nobody's going to say that caffeine is essential to function. Whether you have a bad coffee habit or not, if you don't have coffee, you're not going to die. Okay, it's not essential. If you don't have coffee for 10 years, nobody's going to die, right? We all know this. Next example, Viagra, sildenafil, that little blue pill, right? It has the drug/biological effect of temporarily assisting with erectile dysfunction. It works fairly well from what I'm told, but nobody's going to say that you need Viagra to make your parts down there work, right? People lived before Viagra just fine. So when people say that vitamin A does stuff, it does things. Yes, the so-called vitamin A, like many other compounds, has some definite temporary biological effects. But just because it does stuff does not make it an essential nutrient. If humans can go without eating it for 2 years, it's not a vitamin. The studies I went over show this, and we have more and more people every day avoiding vitamin A with an adequate diet and getting better from their chronic health conditions. So now I'm going to get into the current real-world research, the real-world researchers in the trenches, of which I am one, and my clients are also. This is what's going on now. So we have Grant Genereux. Grant is a buddy of mine. He is the guy who brought my attention to vitamin A toxicity in the very first place. His website is ggenereux.blog, and I highly suggest you read his free ebooks there. He talks about his journey and the science. I mean, it's great. Grant had chronic kidney disease and eczema to the point that the doctors told him he was going to have to get on dialysis or he was basically not going to last much longer. Okay. He figured out, well, he had a theory that vitamin A toxicity was behind it based on his research. And you can read all about how he did that in his books. He has now, to this point, minimized his vitamin A intake from food for 6 years, longer than any of those studies. Okay, 6 years. If it's a vitamin, he should be dead by now, right? If it's really a vitamin and he's 6 years depleted, he should be dead based on what everybody thinks. He should be blind, he should have terrible skin problems, and he should probably be dead. His serum retinol has recently tested at 0.1 micromole per liter, which in the units that we tend to use here in the US, that's 3 micrograms per deciliter. They say deficiency starts at a level of 20 or below-ish, 20-ish or below for adults. So he is severely deficient. The lab, when they ran this test on him, they actually contacted him because they were worried that he was so low. Okay? So in terms of the blood work, this is a serious low number, right? Well, what has happened to Grant since he's been doing this? His chronic kidney disease is gone. He says that his kidney doctors say that he is the only person they've ever seen recover from chronic kidney disease. Fully recover. Not have to go on dialysis. The only person. His eczema is gone. And his health has basically improved in nearly every way. Pretty strong experiment, right? Even if it was only one person. Now we have me. I'm Dr. Garrett Smith of nutritiondetective.org and the Vitamin A Detox Program. I used to have psoriasis. I had some really chronic middle back pain that nobody could figure out. And I had urinary prostate issues starting in my mid-20s. My dad died of prostate cancer. It was pretty concerning to be getting those in my mid-20s. What have I done so far? I have minimized my vitamin A intake after I found out about Grant's work and I started on it. And now I'm kind of the guy figuring out how to get this stuff out of your body the fastest possible. I've been minimizing my vitamin A intake and doing the other things that are in the Vitamin A Detox Program, which are way beyond just not eating vitamin A, for over 2 years at this point. I believe I'm at about 2 years and 3 months. Okay? My psoriasis is gone. My middle back pain is gone. The urinary and prostate issues are gone. My chronic insomnia is gone. And my health has basically improved in pretty much every single way. Okay? Between Grant and I, we are currently aware of about 1,000 plus people restricting their vitamin A intake in an effort to fully correct their chronic disease states. Now, if vitamin A deficiency was a real thing, you would expect people to be dropping like flies on this, right? We are the leading from the front. We are putting our money where our mouth is, and we are regularly seeing people improve their supposedly chronic and incurable or intractable health conditions, including autoimmune conditions. Okay. We have more on this. Animal research. So the supposed classic 1925 study of Wolbach and Howe, I have it linked there, that people say proves vitamin A is an essential nutrient. They killed most of their rat subjects with diet within 9 to 11 weeks. That's less than 3 months, folks. They killed a lot of their rats with their diet. Grant's not dead. I'm not dead. We're not getting worse. But here's something important to note about their study, and I may do a video just taking apart this study, because this is the one that people like to say is it. This is what proved it. Their study had, I looked at the mineral mixture. You can go and look at the paper to see the mineral mixture. There is no zinc at all in their mineral mixture, and there's no reason to suspect there's any zinc in the food they fed the rats. Then the casein protein, which was the main source of protein on the diet, did not contain any taurine. And for those of you who doubt that, go and look up cats and taurine and casein. Go type in cat, taurine, casein. You'll see that when cats are fed a casein diet, they get retinal problems from the lack of taurine in their diet. You may have already been hearing about in pet foods, how there's not enough taurine. And now dogs are getting heart problems and cats are getting all sorts of problems because there's not enough taurine in the pet food. There's also way too much vitamin A. Gosh, those might work together somehow in a bad way, like to kill rats within 9 to 11 weeks. Zinc deficiency, fatal. Do you remember that? Zinc deficiency could be fatal. Taurine deficiency causes all the symptoms that are supposedly vitamin A deficiency. Now, elsewhere, we have polar opposite findings of Wolbach and Howe's. Grant, who lives in Canada, kept a gerbil in perfect health on a vitamin A-free diet for 33 weeks. Another person in Australia kept mice on a vitamin A-free diet for a full year, 52 weeks. This is very different from Wolbach and Howe, right? And we have better science now because they were just figuring out what vitamin A was back in 1925. They weren't really that sure of what it was. So they couldn't say very well what it was in and what it wasn't. Like if you're trying to say, we're trying to figure out what this thing is inside of food and we're not sure what it is and we can't really analyze it because we don't know what it is. We're trying to see if animals need it or not. They don't know anything about what vitamin A is actually in and what it's not because they don't know what it is. There was plenty of vitamin A in that rat diet, probably even toxic levels of vitamin A. Toxic levels of vitamin A plus no zinc, plus no taurine, dead rats in 9 to 11 weeks. Elsewhere, people give mice and gerbils adequate diets and they have no problems. There was the study we went over earlier, 2 studies. And then there's these homegrown studies. This is real science going on, folks. Moving on. So now we're going to revisit this previous research with our new knowledge. So how does this compare to one of the studies I discussed earlier? Here's the quote from that study. In the rats which lacked vitamin A in their diet, there were no special outstanding pathological findings, end quote. There was nothing wrong with the rats that got no vitamin A in the diet. We have here assembled multiple long-term human and animal studies that show that diets that exclude vitamin A do not cause problems. What this means is that the studies claiming vitamin A is an essential nutrient are not replicable because other variations of human and animal diets that don't contain vitamin A don't cause any problems at all. That's sort of important. So if you have people saying this diet didn't have vitamin A in it, but it caused disease, and other people say, well, our diet didn't have vitamin A in it either, and it didn't cause disease, that means these studies are not replicable, and the old ones should basically be thrown out. So what is replicability in science? Why is it important? Quote, replicability means obtaining consistent results across studies aimed at answering the same scientific question, each of which has obtained its own data, end quote. The scientific question here is vitamin A essential? There's not consistent results. Have I not shown that? It is of critical importance that research is replicable because if studies looking at the same scientific question cannot be repeated with the same or similar results, then this suggests an error, either by accident or on purpose, in procedure, like the diet, or recording of the data, which creates massive doubts in the reliability of the research and the conclusions it proposed. Now, did you know that there's said to be a replication crisis going on in medicine right now? Weird. Quote, the replication crisis is, as of 2020, an ongoing methodological crisis in which it has been found that many scientific studies are difficult or impossible to replicate or produce. The replication crisis affects the social sciences and medicine most severely, end quote. So here's the idea. Could somebody go and reproduce the Wolbach and Howe study and feed those rats that garbage diet and kill them too? Yes. Have I shown adequate evidence here of replicability of low vitamin A diets? No, they're not replicating the same thing. We've shown multiple studies here where they showed that vitamin A free diets or low vitamin A diets didn't cause any problems. This means this calls the whole vitamin A as a vitamin, and that there is such a thing as vitamin A deficiency into question, or just debunks it entirely. As we said here, since the Wolbach and Howe study is not replicable in other research studies using different diets that are all free or technically extremely low in vitamin A, here is the likely answer to what is going on from a study I discussed earlier. Quote, it would seem from the foregoing experiment that in cases of xerophthalmia, which occur in human beings, other factors other than fat-soluble A may have to be considered in regard to the etiology and treatment of the condition. What have we demonstrated here today? We have demonstrated that animals don't need vitamin A in their diet. We have demonstrated that humans don't need vitamin A in their diet. We have shown that low blood vitamin A, aka serum retinol in humans, is not implicitly associated with any problems in countrywide populations. We've shown that multiple nutrient deficiencies cause the exact same eye and skin issues said to be caused by vitamin A deficiency. We have basically demonstrated that vitamin A is not an essential vitamin in spite of vitamin A having observable biological effects. It does stuff. I'm not going to argue that. It does stuff. That doesn't mean it's a vitamin. After all of this, if we just called the existence of vitamin A as a vitamin into question or have debunked it entirely, what does that leave? That leaves vitamin A toxicity. That's all it leaves. Vitamin A toxicity is very real. Some of you out there watching it, many of you out there watching it, have vitamin A toxicity. You may know it, you may not. You may be watching this going, I heard about this and it sounds like I might have it. Go and watch my other videos on my Nutrition Detective channels, youtube.com/Nutrition Detective. I have all sorts of videos there showing toxicity. Toxicity is legit. Toxicity can be reproduced. I have multiple papers on eating liver causing vitamin A toxicity. I have multiple papers on, I have a paper on carrot juice causing a rabbit to get vitamin A toxicity and get multiple autoimmune disease in joints all over its body. Okay, this is legit. Food vitamin A can become toxic and is toxic, and supplement vitamin A can become toxic and is toxic. And oh my gosh, the pharmaceutical retinoic acids like Accutane, isotretinoin, retin-A, tretinoin, those are the most toxic forms of vitamin A at all. And yes, your body does take vitamin A that you eat and turn it into the exact same retinoic acids that are called Accutane and retin-A. The research on this is undisputable. You make Accutane and retin-A in your body if you eat vitamin A. Do you think that could be what causes the problems? Because those drugs have nasty side effects. Whether it comes into your body by a pill or whether it comes into your body by carotenoids and it turns into retinoic acids on the way through, it all causes problems. Anyway, that is my presentation for today. Hope you all enjoyed it. And I will see you hopefully in the future on more videos. I was going to do a video, if you didn't know, Bill Gates loves vitamin A. Bill Gates, his favorite vitamin is vitamin A. Did you know that? It's really interesting, isn't it? And then I'm going to do a video taking apart that Wolbach and Howe study and all the problems with it and why it killed rats within 3 months. So I hope you all enjoyed this. I'm Dr. Garrett Smith, the Nutrition Detective of nutritiondetective.work and nutritionrestored.com. And I hope you all have a great day and you learned something. Bye.